Okay, everyone. So now we're going to talk about the nervous systems. This is probably my favorite topic of the whole thing. There is no way that this 20-something slide PowerPoint is really going to do it any kind of justice, but we are going to talk about it in the context of cell signaling and how our nervous system plays a role in cellular communication. So, organization of the nervous system. There's a ton of stuff. There's really two main divisions. One is called the CNS, or the central nervous system, and that consists of your brain and your spinal cord. The other is the peripheral nervous system, which really goes throughout your body. And I always think of it like your peripheral vision is your side vision. So your peripheral nervous system is kind of like your sides or every other part. And your peripheral nervous system consists of a lot of parts. It's pretty much everything that's not your brain and spinal cord. And there's a series of players. There are sensory receptors, and we've learned what receptors are. They are the things that are going to receive the sensory information. So an example would be the receptors on your taste buds. Think of what they were feeling when you consumed apple cider vinegar. Then we have sensory neurons, which really sense information. So these are triggered. For example, sensory neurons might live in our fingertips. So if we were to touch a hot stove, then those sensory neurons are activated and it will trigger information to be sent to your central nervous system. Your brain needs to be told that, hey, I have a hand on a hot stove. Motor neurons are kind of the other way around. Motor neurons are the ones that talk to your muscles to produce motor movement. They could also talk to your glands, but the idea would be like a motor neuron would tell your muscles to move. It would tell your brain, it would tell your arm or hand to move away from the stove. An interneuron is something that's in between. It connects the sensory neurons and the motor neurons. And we'll talk about how the whole thing, like it's called a reflex arc. We'll learn it later, but let's say I put my hand on a stove. The sensory neuron is going to tell my brain what's going on. An interneuron is going to connect that information to a motor neuron that's going to say, hey, move your hand. Nerves, as we know them, are really bundles of neurons. A neuron is an individual nerve cell, and those come together and work together in a nerve. And oftentimes they contain a bunch of different types of neurons. So it's not just all sensory or all motor, but really it's a combination of both most of the time. Here's just a visual representation of how the nervous system is broken down. There are sensory divisions. So afferent really is going to the brain. So it's transmitting information from the sides or the periphery to the brain. And there's different subdivisions within there. Somatic just means body. Visceral has to do with your organs. But in addition to sensory, we also have motor. Motor is called efferent. Exiting the brain is how I like to think of it. It basically is sending information to actually move. So somatic, motor is our voluntary movements. So we voluntarily move our arms and legs and things like that. We also have autonomic, which I always think of as automatic. These are the things that we don't really consciously control. It just happens. Like we don't control our heartbeats. We don't control our esophagus and our intestines for pushing food through our body. And we don't really normally control our breathing. If we think about it, probably, but the idea is it just kind of happens. It's automatic, and that's the autonomic nervous system. And it's motor because it's connecting the actual physical reactions. You don't really need to know the super crazy specifics of all these. We're going to be talking about it in terms of signaling, but it's good to have a background. Okay, so here's kind of just what that looks like if you're cartoon looking at the CNS, which is again your brain and your spinal cord, and you can kind of see that down the spinal cord there's all of these spinal nerves. You have your sciatic nerve coming down here, which is why oftentimes it can give you, leg pain or from your sciatic nerve, and it kind of looks like a horse's tail down here. But you have all of these spinal nerves radiating out that go into the periphery. We also have ones in our face too. Those are all of our cranial nerves, which is why sometimes people with stroke or specific conditions can have partial facial paralysis. So here's kind of the overall piece that I was talking about. So there's some kind of sensor, and there's a sensory neuron or sensory input that will go to the brain, and then interneurons will communicate information and send it out to a motor neuron to actually do some kind of output, or in this case, effector. And the central nervous system is the one doing the integration piece, whereas the peripheral nervous system is either getting the info there or getting it back out to the muscles. Your peripheral nervous system can either be somatic, as I mentioned, so these are the voluntary, generally conscious things that you're doing, whereas the autonomic are the things that you don't really control. So this is related to breathing and or heart rate, pancreas secretions, as well as like digestion. So I'm not really going to make you know the nitty gritty of each one of those, like I said. Just kind of generally knowing parts is helpful sometimes. And here's what that looks like. So in terms of your body, we have sympathetic. Sympathetic relates to, I always think of the fight or flight response. Sympathetic is fight or flight. So think about all of the things that happen to your body when you get crazy scared. So you are either going to fight or you're going to run. So what's going to happen? Your pupils are going to dilate so you could see in the dark. You are not going to salivate because you are not thinking about food at the moment. You are going to accelerate your heart rate. You're going to, again, cool all the things that have to do with food, eating, and digestion. You're going to inhibit your stomach. You're going to inhibit the pancreas. You are going to release glucose so that you can have energy if you need to run, those kinds of things. You're going to inhibit emptying the bladder so that you don't have to pee. All of those things. You are going to be ready to fight or run. Whereas parasympathetic is what happens after you have like a giant Thanksgiving meal. You just want to sit on the couch and digest. So your pupils are going to constrict. You don't need to have them open to run. Your salivary glands are going to start doing their thing. The stomach is going to do its thing. The pancreas is going to do its thing. The gallbladder, all those digestive things are going to go. Your heart rate's going to slow down. You might have to go to the bathroom or whatever. You're just going to kind of chill out. So here is one of the key pieces, I think, of what you're going to have to know in terms of the nervous system is the general neuron and how it actually communicates from one cell to another. So this is a neuron. It has a couple of major pieces. So the first one is called a dendrite, and that's where these little branches are. That is where the information is coming in. So the stimulus, whatever it might be, is going to land on the dendrites, and all the little receptors are on those dendrites. They then communicate information to the cell body, which is like the main part of a neuron. And within the cell body is where the nucleus is. Kind of looks like a little one-eyed monster. But the idea is all of these dendrites here are collecting information, and then they pool all of that information together and send it down the axon. It's a one-way travel down the axon to the axon terminal, wherever it ends. And this is really a zoomed in picture here on the left of what that looks like, where those little terminals are essentially going to connect to the next neuron and its dendrites so that the dendrites can pick up that signal and then transmit it down its axon, down to its axon terminal and on to the next cell. So in these axon terminals, you can see there are little vesicles, which we have learned about. They're like little Ziploc bags. In this case, they're holding neurotransmitters. These are the signaling molecules that will attach to and cause a response on receptors of the next cell. So these little neurotransmitters get released. They travel across what is called a synaptic cleft. Cleft is an opening. And then they can land and trigger the receptors on the next neuron. And if it's enough signaling, it will then trigger that nerve cell to talk to the next one. And you can see in this picture on the right here, the nuclei are stained in blue and the rest of the neuron is actually stained in green. So you can see like here's a cell body and then the axon. You can see it's not super clear all the time which one is an axon and which one is a dendrite. They're kind of all tangled together, but you get the idea. Okay. This highlights a lot of what I mentioned before, but just to recap, a neuron, the major piece is the cell body, and that's where the nucleus lives as well as other organelles to kind of sustain the neuron. The dendrites are the branches that are receiving information, so they have the signal receptors to receive information. The axons transmit messages away from the cell body to the axon terminal and on to another cell. What I haven't yet mentioned is called myelin sheath. And this is an insulation around the axon, and I'll show you, there's a picture of it in a minute. But it's insulation that wraps around the axon where it will help make the message go faster. And think about our brain and, the amount of brain cells that we have. Our reactions have to be very quick. So myelin sheath helps that happen. And when we don't have it, our nerves can't fire as fast as they need and we can't perform the functions that we need. The synapse is where two neurons come together. So it would be the axon terminal of one and the dendrites of another. And that space in between is called the synaptic cleft. A neurotransmitter is that signaling molecule that travels across from one neuron to another. And then another thing I didn't really mention before are glia. Glia are little helper cells. They're like the godmothers of neurons. They help support and nurture the neurons to make sure that they have all of the nutrients that they need to survive. And one example are called Schwann cells, probably because some guy named Schwann figured this out. But Schwann cells are the cells that make the myelin sheath. They wrap it around the axon so that they can function properly. And here's a picture of what that happens to look like. So you can kind of see that there's insulation wrapped around the axon, and that insulation, keeping in mind a Schwann cell it's an actual cell. It's got a weird little nucleus here, and then the whole thing wraps around, helping the signal go faster. And in between these little sheath areas are called the node of Ranvier. And what ends up happening is the neuron signal basically skips over the myelin sheath and just hops along the nodes to make the signal go faster. Another key important part is how the neuron actually signals and how some get turned on and some don't. And this is what we call a membrane potential. And we have learned about channels in the membrane, and we have learned how certain molecules move across. In the case of a neuron, it really has to do with a difference in charge, whether it's positive or negative, and how much so. In this case, there is a sodium-potassium pump, as well as a sodium channel and a potassium channel. So channels, if you remember, just open and allow movement where diffusion would like to go. So channels just open and there's free movement. For example, the potassium here, which is a little diamond shape, will go from high concentration inside the cell to low concentration outside the cell. That would be an example of a free moving channel, whereas sodium, if it opens, sodium is going to rush in because there's a high concentration outside, a low concentration inside. The pump, because it's called a pump, should tell you that it is requiring ATP. It pushes those things across the membrane where they don't want to be. So it actually ends up pumping 3 sodiums out for every 2 potassiums in. And you can kind of see it's got 3 bumps on the left for sodium and 2 diamond halves on the right to pump potassium in. And so because it's going against the concentration gradient, it requires ATP, but its goal is to maintain a negative potential. The neurons want, in this case, when they're off, to be negative. Okay, so this brings us to what we call an action potential. That is when the nerve fires. There's some kind of nerve impulse, and the neuron has received enough stimulation from the dendrites and the receptors on those dendrites to trigger a true action potential or a true impulse down the axon to stimulate the next neuron. So as I mentioned, when a neuron is at rest, it has something called a resting potential. It likes to be negative. And the way that the pump works is that it has sodium on the outside and potassium in high concentrations on the inside. There is also the voltage-gated sodium channel. Remember the channel that I mentioned where sodium would then free flow in is normally closed. It is voltage gated, meaning that at a particular voltage, it will then actually open. But under resting conditions, the pump is maintaining the concentration gradients and the sodium gate is closed. When a nerve impulse happens or an action potential happens, the stimulus causes that potential to change. The membrane is going to what we call depolarize. And there's a change in voltage, which I'll kind of explain in a minute. But that opens up the sodium channels. And then there's going to be a rush in of sodium. And when that happens, that rush in of positive charge totally changes the negative that was in there before, and that will trigger a response. And it's important to know that an action potential is going to be an all or nothing. You either reach the necessary threshold to make this magic happen, or you don't. It's really a function of, do you get to the point where you open those voltage gated channels? If you do, there will be an action potential or nerve impulse. And if you don't reach that threshold, then you won't. Those channels will remain closed. So overall, what happens is sodium channels open, and then that causes sodium to rush into the cell. And then after that point, potassium channels open, which are also voltage gated, and then sodium starts to leave the cell because it's going to diffuse out of the cell from high concentration to low concentration. And eventually, the sodium-potassium ion pump will have to put everything back against the concentration gradient that it was when it was at rest. So here's roughly what this looks like. This, I would say, is a very important slide. So one is the resting state. So inside the cell is highly negative. Outside the cell is positive. You have your special pump that is making sure to keep the sodium concentrations high on the outside, potassium concentrations high on the inside, and the channels are closed in this case. Then depolarization happens. So the nerve cell has received enough stimulus to open up the sodium channel. So this sodium channel will open up, and then all of a sudden we will have an influx of sodium. And then that's enough to trigger other sodium channels to open. So that's kind of why it moves down the axon. It's this triggering cascade. And in terms of a graph here, just to kind of highlight, our resting state is somewhere around like negative 70-ish. So it's happy at that point. And then 2, some of those sodium channels are triggered. And then by 3, if you reach that certain threshold, if enough sodium channels are triggered and you move enough positive in to the cell so that it's now at a negative 50 membrane potential, it will reach that threshold and it will shoot up. All the sodium channels are opening. Tons are rushing in. And then what happens is at this super high potential, the potassium channel opens. So at this voltage, it triggers an opening of the potassium channels, and then potassium can start rushing out. So now you have positive leaving the cell, and that will cause the inside to become more negative, and you can see that it goes down again. And what happens is it actually overshoots a little bit. I guess this slide is calling it undershooting, but it goes below because all these things are open. And in the falling phase, the sodium channels begin to close. So the sodium channels have closed. Potassium is still rushing out. You are now pumping all of this positive out that you actually go past what your normal is. And then you can kind of eventually get back to normal, to the resting state that you were beforehand. So looking at this graph and knowing why it's happening is extremely important to nerve signaling. And here you can kind of see how that goes along an axon. So if you have an action potential, normally it's going to cause an influx of sodium, which causes the inside to suddenly turn positive. And then it kind of acts as a domino effect. This positive is enough to trigger an action potential in the next set of sodium channels, and so on and so forth. And then as it follows, the potassium channels open and reestablish that negative potential that was there before. In terms of the myelin sheath, as I mentioned, it helps increase conduction. So the Schwann cells create this myelin sheath, and then there are nodes that are exposed in between. So the action potential actually skips over those insulated regions and triggers the node that will then trigger the next node and the next node. So it's called saltatory conduction. Don't know why, just is. But it triggers those sodium and potassium channels as you move down in those node regions. Okay, so this part should be a little bit familiar in terms of cell communication. Neurotransmitters are the signal in this case. They are released into the synapse by what we call a presynaptic membrane. So this, or a presynaptic cell, meaning before the synapse. There's also a postsynaptic cell, which is kind of the receiver. And in this case, we have vesicles that will be releasing or dumping their signal, the neurotransmitter, into the synapse or the synaptic cleft area. Those neurotransmitters travel across and land on receptors on the postsynaptic membrane, so after the synapse. And when they bind, in this case, the neurotransmitter is acting as a ligand and attaching to the ion channel. They're called ligand-gated ion channels, and it will then trigger a response in the cell that comes after it. So in this case, the ligand binds to a receptor that will cause a quick influx of sodium and efflux of potassium, and that might be enough to totally trigger an action potential. In the next neuron, anyway. There are a ton of neurotransmitters, but important to know is that they are chemicals. As I mentioned, they are released into the synaptic cleft by vesicles. So this is exocytosis. They diffuse across the synapse. They stick themselves to receptors. It could be another neuron. It could easily be a muscle cell or a gland cell. And most of the time what happens is those chemicals are either broken down by enzymes or they get taken back up by surrounding cells, because you can't have a signal on all the time, triggering receptors all of the time. We could not handle that. And neurotransmitters acting as a ligand or a signal, we've learned they can either be excitatory, so they are going to make things go faster by causing depolarization or action potentials of the next cell, or they could be inhibitory. They could actually slow transmission by hyperpolarization, which is a fancy way of saying make the neuron even more negative so that you have to put in more input to reach that threshold and actually create an action potential. There are a ton of different examples. You don't have to know them specifically, but I just wanted to introduce you to a couple of them. So acetylcholine is one of the most common ones, ACH. It stimulates muscles and learning and memory and all kinds of things, but usually muscles is kind of what we learn it as. Epinephrine is also known as adrenaline. That's the one that gets released when we trigger that fight or flight response. Same thing with norepinephrine. Those are usually all stimulatory. We have dopamine that gets released when we are happy or excited. These are the reward neurotransmitters. So whatever you like the most, if there's like a, I don't know, if you're addicted to candy or other things, then that's the reward center. Some people could even have dopamine released upon playing video games or binge watching or Snapchatting or whatever. Loss of dopamine is one of the problems with Parkinson's disease. Serotonin has to do with well-being and happiness. Low levels of serotonin have to do with depression, which is why a lot of depression medications like Prozac, for example, stimulate serotonin neurons. And then we have GABA, which is a primary inhibitory neurotransmitter that is affected by alcohol. So alcohol inhibits a lot of thinking neurons, go figure. All right. So I think it's the last thing that I'm going to mention is really reflexes. And I talked about it earlier in the sense of sticking your hand on a hot stove and what that means. But it's a simple automatic response. You really don't necessarily think about it. Like there's not a deep brain thought process. A reflex arc is an unconscious thing that happens. So the receptors on your hand, for example, might sense the heat that is triggering or creating an action potential in a sensory neuron. The interneuron is located in this case in the spinal cord or the brain stem and is going to take that information and turn it around to a motor neuron that will then trigger some kind of response, either in a muscle of your hand to like move it away, or it could be an organ, something like that. That's called a reflex arc. And here's just an example. So if you're at the doctor and they take that little mallet and hit you to do a knee-jerk reflex, the sensors in your knee are triggered, will then go to your spinal cord, interpret that information. So your quad interprets that information, sends it to your spinal cord. This would be an example of a vertebrae here. And then a motor neuron would then be triggered to actually cause you to do that kick in the hamstring muscle. All right. Finally, just understand the brain is a pretty complex thing. So we have specialized regions that do different things. And this isn't a foreign idea. Even an embryo in 1 month has all kinds of different crazy areas. But if you've taken psych, you're going to be a little bit more familiar with these things, or HBS. But we have a forebrain, we have a middle brain, and we have a hindbrain as 3 major areas. And here's kind of what that looks like in detail. So the cerebrum is the big guy in kind of yellow. The thalamus and the hypothalamus are in here. The pituitary gland is this little guy right here. That's where most of the endocrine signaling comes from. We also have a midbrain and then the hindbrain, which includes the pons, medulla oblongata, and the cerebellum, which is where your balance comes from and coordination. And then you have the spinal cord. In terms of important things to just remember, our cerebrum is in charge of the signaling necessary for information processing. So our voluntary movement, our learning, our motion, all of those things. Our brainstem is the basic part. That's controlling kind of our autonomic functions, our breathing, our heart and blood, our digestion, our swallowing, and so on. And the cerebellum, as I mentioned, is coordination of movement and balance, our motor skills. So depending on the signaling that's happening and where it's happening, we get different responses in our body. And then that concludes the neuron system or neural systems.